I'd like to ask you to take a seat, and we will start with the next session. Okay, well, I'm very excited and very honored to announce the next speaker for you. It's Professor Richard Lieber. He's the Chief Scientific Officer and Vice President at the Shirley Ryan Ability Lab, formerly and famously known as the Rehabilitation Institute of Chicago. And we're so happy to have him here today to provide this keynote. He's a true authority in the field of muscle morphology worldwide, and has contributed to our knowledge of the muscle in CP in a very big way. He's been publishing on this subject since the 1980s, so over a very long time, and providing us with a lot of knowledge on this very important piece in the puzzle of motor dysfunction in CP. He's been very active already in the pre-symposium that was loaded yesterday, and also a symposium today. But he's promised me that he will provide you all with new information during this keynote. So I'm very happy. Also a little bit afraid, because I have to say the more I hear about all these muscle properties, the more confused I'm getting as a clinician about what to do. But maybe you will help us, and I have all confidence in that. This is a very special lecture, because it's not just a keynote, but it's the Fondation Motrice keynote that we're going to listen to. And the Fondation Motrice has awarded Professor Richard Lieber this honor because of his important work in the field of muscle morphology in CP. And I think that the professor will recognize the importance of this French award, also in recognition of important predecessors like Tardieu, who has been publishing on the muscle in CP since the 1950s. I would like to ask Dr. Nathalie Jeunesse from the Fondation Motrice to present the award.
So thank you very much. I am representing La Fondation Motrice. So what is La Fondation Motrice? It is a foundation dedicated to the research on cerebral palsy through a yearly call for projects. I just want to say to you that we will have a more important call for projects in June, so we will be aware of that. And we are very happy to support the work that is done by the EACD, and we want to honor Professor Lieber for this lecture and for the work that has been done on muscle and in patients with cerebral palsy. So we are very pleased to give him this award.
Beautiful. Thank you. Thank you very much. That's beautiful. Yeah, sure. Yeah. That's beautiful. Thank you.
With a back to the
all the people.
Audience. Back to one people. Yeah, back to you.
Okay.
Important.
That was awkward. Feels like the wedding, right? Just go have the reception. So thank you very much for the award. I appreciate it very much. It's a beautiful award. You're going to have to take a look at it. Just don't take it with you. I'm excited to do this because I came into the cerebral palsy research area explicitly a little bit late. I'm showing this picture because Dave Sutherland, an orthopedic surgeon in San Diego, in 1984 invited me to be the presidential lecturer at the AACPDM meeting in Washington, D.C. And I remember it because I'd never been a lecturer invited somewhere. And I came there and he asked me, when I showed up, he said, "Well, do you have a tie?" And I said, "No." I was a graduate student. And he said, "Well, I'll get you a tie." He goes, "Do you have a coat?" "No." "I'll get you a coat." So I awkwardly went into this meeting with a weird tie and a weird coat and to a Marine marching band, and I literally talked to all these really nice people about frogs. And I know half the people there were thinking, what is this guy doing here? But I gotta tell you, today I have some frog data to show you, and I hope you'll see that it's relevant, and hopefully it will be interesting. Jan Freden is a hand surgeon from Gothenburg. Now he's in Nottbil in Switzerland, and Hank Chambers is an orthopedist in San Diego. They're the two surgeons I've worked with the most over the years. And Lucas Smith is the PhD student, and I'm using a lot of the data from his PhD thesis. So, a frog guy. Frog people learn early on that muscles look like this. I'm sure you've seen this picture a million times. For some reason, we all have to learn in medical school, graduate school, therapy school about A bands and I bands, right? Did everybody learn about A bands and I bands? Yeah, and who cares, right? It's on every board test. It's just one of those things. So I put it in there. I always say it's muscle malpractice not to say A band once during a lecture. So I said it. But the important thing about it is, of course, that the A band is where the motors are in skeletal muscle. They power muscle contraction. And what's most important is that the A bands and I bands together make this machine, which is known as a sarcomere. And the sarcomere is the fundamental force producer in muscle. So I was born and raised as a frog, single muscle fiber, sarcomere mechanics guy. It wasn't funny. It was hard. I grew up on this curve, and I worshipped at the feet of Andrew Huxley in London, who showed that the structure of the filaments actually gives rise to the mechanical properties of the muscle. And as you know, there's an optimal length for force production in muscle when the actin and myosin are at their optimal length. And at long lengths and short lengths, muscles produce less force. This was always intriguing to me, and my PhD thesis was the mechanics of the cells and how it might be implicated in frog hopping. About that time, I had a guy on my committee who was a functional morphologist. So he studied, bee flying and squirrel digging and porpoise swimming and elephant water shooting, and he just studied everything in animal morphology. And he asked me, what difference does it make to a frog that his sarcomeres are like that? And I thought that was a good question. And what he was really asking was, in terms of physiology, where is that muscle designed to even work? And why should it work where it does work? And of course, I couldn't answer the question because I had always taken the cell out of the frog. So as I was finishing my thesis, I moved from Davis down to San Diego. I started working with hand surgeons, and I started seeing this sarcomere length tension curve has profound surgical implications, because if you're a surgeon and you overstretch a muscle and it ends up over here, that's really bad news, because the muscle could be, 50, 80% weaker simply because it was overstretched. Or vice versa. What if you didn't put it in tight enough and it was over here? And we started having these discussions, and again, it sort of came out, where do these muscles operate? So we went back to the basics, and the reason I point out frogs are the basics is frog is the only muscle for which we have X-ray diffraction level filament data and single cell physiology data to know that this relationship is true. For mammals, we're sort of inferring that it's true, and it probably is. But I decided to start my studies in frogs with kind of the gold standard. So we just call this like a frog Cybex machine. So these are just pieces of a frog, a pelvis, a femur, a tibia, and a muscle. But the nice thing about this system is, unlike most of what is going on in this meeting this week, this system is very well defined. It's a muscle, joints, and measuring tools. So we could take the joint and rotate it and just see what the range over which sarcomeres would work, and we were surprised to find that it's actually a huge range. This is work that Jenny Boakes, who later became an orthopedic surgeon, did in my lab. So you can see here with the knee nearly fully extended, sarcomeres are at about 3.6 microns. And if you're a sarcomere geek, like I am, 3.6 is the length where they barely generate force. And if you look at full flexion at around 2 microns, that's the length where they generate maximum force. So if your goal was to do rehabilitation in frogs, and the frogs hop into the clinic with problems, and you start treating them, you have to take into account the fact that the design of this particular muscle has a huge force variation throughout the range of motion. And if you put the whole system together, and I'm not going to go into the details, this is the take-home lesson. This really had a big impact on me for the following reason. The joint moment here, and this is explicit data. I'm drawing it here, but it's real live data. Nothing here is modeled or assumed. Okay, we measured all of these, and they worked. And that's a good story in itself. But anyway, the yellow line here, the joint moment, is basically strength. And so the first thing to remember when you're measuring patient strength is that strength, which is the product of the muscle force and the mechanical advantage of the joint, is a combination of two things, muscle and joints. And if you notice here, if you wanted to make this frog the strongest, you know what you would do? You would actually exercise the frog at near full flexion, where muscle force is maximum. Because muscle force, muscle stress, when muscle stress is maximum, the strengthening input is maximum. How many of our patients do we ever exercise or treat in joint positions where they don't generate much torque or force? So that's where you start to, you really have to think critically and dissociate the tissue level responses or the tissue level performance from what can be observed in a human or in an animal. So that's one point. And the other thing is this joint moment isn't the position where the muscle generates its maximum force or the mechanical advantage of the joint is the maximum. It's just an intermediate. And this mix of muscle and joint properties is different for every joint in the animal and for many joints in humans. So now, when I think about the design of the musculoskeletal system in humans, one of the things I think of is the ratio of the length of muscle fibers to the size of moment arms. And you can look at wrist, elbow, shoulder, hip, knee, we've done all these, even ankle. And each system sort of has its own design. And when you're treating it, you sort of have to think critically about it. And we talk a lot to therapists and physicians about that. So fast forward a few years, and so this is 88. About that time, I also met Jan Friedan. He was doing a postdoc in San Diego, and he was doing some of these tendon transfer surgeries. And essentially, we had learned a bunch of things in animals. We just wanted to start learning them in people. So he and I essentially modified a laser pointer and something that would be sterilizable. And we exploited the fact that muscles have this A-band pattern. So this is what I'm showing here. The band in the inset is the A-band that I showed you previously. And a nice property of this muscle is that it looks like a crystal to laser light. So if you shine the laser light into the muscle, you end up getting the diffraction pattern that's shown here in red. And that diffraction pattern can be converted into sarcomere length. So how cool is that? You can go into the operating room, put a gizmo into a person's arm, move the joints around, and measure sarcomere length as you rotate people's joints. Super interesting stuff. We did a bunch of those studies, but I'm just going to give you a couple of the take-home lessons now because I want to talk about, ultimately, wrist flexion contractures. But this is a wrist muscle. This happens to be extensor carpi radialis brevis, ECRB, a prime extensor of the wrist. Wrist joint angle, in the negative angles here are flexion angles, and the positive angles are extension angles, and here's the vertical axis here. So this is only 5 patients, but trust me, at the time it was heroic. In fact, we got these data, and we went directly from this experiments. We were up in northern Sweden, Umeå, and then we went to a meeting in Liverpool of the Society for Experimental Biology, which is a bunch of lizard people and frog people and those kind of people. And we started showing these, and they kept saying, they go, wait, what animal is this from? We go, it's from humans. And I swear, the response was, well, who cares about humans? I mean, is it a roach? No. Is it a locust? No. Is it an ant? No, because these are all these comparative mechanics guys. So we even said, well, we sort of care about people. I mean, even frogs. I really never worried too much about healing frogs in my lifetime, but anyway. Okay, but the take-home lesson is, for this muscle, again, ECRB, with the wrist flexed, you can see by the cartoon, the sarcomere lengths are in the 4 range, which is almost no force, and it's with the wrist fully extended that the muscle generates its maximum force. So, and I'll tell you in general, that's sort of what we see, that joints in their extreme configuration, full flexion or full extension, tend to be maximum force producers. And you can see from this little inset, the optimal length for this muscle is around between 2.5 and 2.7. So mostly that muscle operates at lengths longer than optimum. And if you're a design person or a mechanical engineer, you think that seems like a bad idea. Again, why would God or evolution design a system so that it wouldn't produce its maximum force? So, of course, we asked the same question, and we studied all the 5 prime movers of the wrist. This was work with Greg Loren, who was an orthopedic surgery resident. And The experimental data combined with some kinematics and modeling yielded this human wrist joint design. The basic idea being the flexors tended to operate at short lengths, the extensors tended to operate at long lengths, and our interpretation of why that was a good idea was that as you go from flexion to extension, the flexors get longer and stronger, and the extensors get shorter and stronger. But the key is, when you make the calculations, that there's balance. There's wrist flexion extension balance throughout the range of motion. And the design constraint we thought about for the wrist was, and you know this when you're a surgeon, is providing a stable base for finger movement. And in fact, you can model the system differently. You could put all the muscles at their optimal length at let's say, neutral wrist joint angle. But then force flops off in one direction or the other, and it becomes a little trickier to control. So another take-home lesson here was, when you think about motor control of, let's say, wrist joint stabilization, there's actually intrinsic mechanical stabilization built into the wrist joint. And when you look at why, if you want to read this paper, it's really a sophisticated combination of the lengths of the fibers, the sizes of the muscles, the nature of the kinematics at the carpal joint, and it all adds up to this beautifully balanced system. So in the reconstructive surgery, we focus much more on balance than we do on, how surgeons say, power. That's a long way of saying we had a lot of experience with the wrist by the early 90s. And I'd like to tell the story that we were in Gothenburg by now, and there we were operating right next to a OR that was doing a lot of CP operations, and these were kids coming in with mostly with wrist flexion contractures. And of course we knew that the flexor carpi ulnaris is very tight. It's often bowstringing, and the procedure for relieving the flexion contracture is to cut the tendon and allow the FCU to retract and then open up the wrist a little bit, and sometimes the fingers. So the head of the OR was Ann Nachemson, who many of you may know. She's a great lady. She ran the hand unit. And she said, you guys really ought to study CP. And we were already kind of having our minds blown by how complicated this was. And we were interested in the problem, but we thought it was so complicated. We agreed to do it, but we just didn't think there, sometimes you just do stuff because you should, and she was giving us lots of resources, and we felt obligated slightly, but we didn't really know what we would do. We just thought we would do it. But I remember we put our laser into this first kid, this boy actually, and when you're a true nerd, you can think in inverse space. So remember this slide I showed you, this diffraction pattern? If you're a real nerd, the width of those the lines, the intensity, how they go from, this is 1, 2, 3, how the intensity drops off, you just have it programmed in your brain. When we stuck the laser in this boy, we had 6 lines super close together and knife sharp, which means that in this particular kid, he had a very shortened muscle. Well, the muscle was under tremendous tension, but the sarcomeres inside the muscle were hugely stretched. And that was really a turning point for us for a couple of reasons. One is when you do science and you get an unexpected answer, you're usually right, because most of us are kind of weenies when we design experiments, and we design them to kind of confirm what we're already thinking. So when you get something unexpected, it's important and you have to pay attention to it. And this was true. I remember I stuck the laser in, I looked at, we're wearing masks, I looked at Jan, and our eyes were both big like this, because we both knew what it meant from diffraction. I wrote that home to my wife, I faxed it at the time. And she goes, oh my gosh, it sounds like a romance novel. You looked at each other's eyes? Sort of looked at each other's eyes. Anyway, it was a bromance, an early bromance. But we did. We both knew what it meant, and the data were really clear. This is why, it's not a subtle difference. So on the vertical axis here is sarcomere length again, from spastic wrist flexion contractures versus, these are radial nerve injury patients. We don't typically cut into normal people when we're measuring sarcomere length, but these were patients having radial nerve injury-based tendon transfer. So their FCU is normal. It's being transferred into a wrist extensor. And the reason this is so important is if you look at the length tension curve, you see where those two numbers are. So those are very different, right? 3.5 compared to 2.4. That means from a sarcomere force generation point alone, this muscle is generating maybe 10% of its maximum compared to almost maximum for the normal muscle. That's a big deal. And that alone puts the muscle at a huge disadvantage in terms of force generation. Now, this is one data point from one piece of the muscle. You can still argue maybe there's other stuff going on in the muscle. I can give you some reasons why I don't think that's true, but it's an unprecedented adaptation. I mean, we published this paper in Muscle and Nerve, and it was very hard to get published because people just didn't, they know muscle, and they know it's not, muscles don't do this. Muscles can change sarcomere number, as we talked about this morning in the muscle talk, but they don't get stretched out. They always re-optimize if they do it in animal models. So you stretch them, they add sarcomeres and they get short. You shorten them, they subtract sarcomeres and get longer, but they always end up around their optimum. For a muscle to end up at 3.6 microns is totally unprecedented. And we did this in about 2000. There's no other animal model I've ever seen that does it, and we've tried to make other animal models do it. So I think of this as a uniquely human issue, and it was this experiment that really pushed me to do stuff in people and skip the mouse stuff. So, somewhat later, Margie Mathewson, when we were really trying to directly address this issue of sarcomere length, fascicle length, et cetera, did a follow-up study in the soleus muscle. This is with Hank Chambers patients, where she took ultrasound of the soleus muscle fascicles, which are up around here, so we could measure fascicle length. But then we could take those kids were being wheeled into the OR, we could take them into the OR, and from the same area, we could measure sarcomere length. And I show this for a couple of reasons. So we found, maybe not surprising, that the fascicle length was similar. So kids with CP and typically developing kids who were having other shank limb surgery had about the same fascicle lengths. But as I showed you previously, the critical thing was that the sarcomere lengths in the kids with CP was dramatically longer. And so if you put if you can get your brain around fascicle length and sarcomere length, the fact that kids with CP have longer sarcomeres that are stretched out to the same length means that their fascicles kind of look like this. They're stretched out, the sarcomere lengths are very long compared to typically developing, and importantly, those two fascicles look exactly the same in ultrasound. So I'm just cautioning this audience that I know muscle ultrasound is a big deal. It's a very blunt tool. It's a great way to see how fascicles are oriented in a tissue. It's a great way to measure things like pennation angle. It's a very poor predictor of function. And we can have that discussion if we want to. So the title of this talk is What's the Matter with Muscle and CP? The first thing that's the matter is that serial sarcomere number is dramatically decreased, resulting in stretched sarcomeres. And of course, the first implication of that is weakness, because sarcomeres are long. But the second implication is tons of tension. I mean, when when you stretch a muscle to a sarcomere length of 3.6, it's under a tremendous amount of tension. So that's partly why the joint is deformed. Okay, we started asking questions about what is this tissue? Both Jan and I started focusing on it, and it was not previously an area of focus. We did studies where we would take biopsies during surgery. I was born and raised as a single muscle cell biologist, so I would take one cell out of these kids. So under a microscope, you take the biopsy, you dissect down to one cell. The cell is maybe 2 or 3 millimeters long, maybe 100 microns in diameter or less. And you could do entire physiology experiments on these cells. And I thought, well, now we're going to kill it because we have a huge competitive advantage. We can do all this muscle physiology on these cells. And unfortunately, after many years of muscle physiology, it turned out the cells were really normal. They generate the right amount of stress. There's a stress, 250 kilopascals is kind of the gold standard for muscle. They were right around that. Their sarcomere lengths were normal in fibers. And we were really disappointed when all was said and done, because we measured in the OR that the muscles were stiffer. So what was going on? And we thought, like, it sounds stupid in retrospect. We go, we have the whole fiber here. We have the perfect area measurement. So we know stress here, and it's this. But we measure stress in the OR, and it's that. What are we missing? Like, what's the difference between a fiber and a whole muscle? I mean, that sounds stupid now, but we were just thinking, like, we thought we had the whole system. Well, the problem was shown right here. I mean, muscles are fibers and all the stuff in between them, right? Extracellular matrix. And so we started testing bundles of fibers, and that's when it became really obvious that the major defect in the tissue apart from the sarcomere, this intraoperative stretch sarcomere stuff, or in vivo stretch sarcomeres, the major problem with the tissue was extracellular. And we measured its mechanical properties. There's more of it. You can measure it histochemically, as shown here. This happens to be an antibody for laminin, which is an extracellular matrix protein, but it looks the same for laminin, fibronectin, collagen. You name the extracellular protein, it'll be there. The left panel is typically developing kids, and the right panel is kids with CP. The top is cross sections, and the bottom is horizontal or longitudinal sections. You can just see there's more stuff in between the fibers. Now, this is also a very blunt tool. It's hard to see what's going on. We did a lot of biochemical analyses to try to follow up on these immunohistochemical analyses, and I'm just going to boil the main result to one for you here. So the upper left panel is the modulus, or the normalized stiffness of a bundle, for two different muscles. These are hamstrings, semitendinosus and gracilis, and kids with CP and slightly older kids who are having ACL reconstruction. So as you may know, Hank also does sports medicine. So it's a really nice comparison. Another lesson we learned the hard way is you can't compare arm muscles to leg muscles. You can't even compare one leg muscle to another. If you're talking about, let's say, CP versus typically developing, it better be the exact same muscle. So it took us a long time to figure that out, and to figure out that some of our early results were muscle differences, not necessarily disease differences. But these were good comparisons. Semitendinosus and gracilis from kids about 10 years old with CP to kids about 13 or 14 years old having ACL reconstruction. And it's kind of a fulfilling result. You see the moduli for CP muscles is stiffer, and the collagen content is higher. So we got really excited. I mean, we wanted to maybe have a biomarker for stiffness. Maybe this could be used as a diagnostic. Maybe we can come up with new ways to measure it. The problem was this, and I'm partly doing this as a logic test, just because there's increased bundle modulus and increased collagen content, and they go in the same direction, doesn't mean they're connected with each other. And if you actually take these exact same data and regress the stiffness of the bundles on the collagen content, you can see it doesn't look very good. It's kind of a shotgun blast. So it's not, there is more collagen and they are stiffer, but more collagen does not equal stiffer. It's kind of bad news. You were talking about being confused. You know, I'm used to it because, I mean, when you're a scientist, you're used to being confused. If you're doing good science, you should be getting data you don't really understand. And you should be taking enough data points around what you're doing to show that you're wrong or you're right. Another thing we need to learn as a group is to do positive controls and negative controls for every experiment we do to make sure we're on the right track. But this led us to another area which is ongoing in the lab, which is trying to understand if it's not just the amount of collagen, what is it? And so I had a new student starting, Allison Gillies, and she and I started reviewing the literature on the extracellular matrix of muscle. And the good news is when you're sort of leading the edge on the review and in the literature, usually you have friends at journals, you say, hey, can we write an invited review? And the editor said to me, well, if you're asking me if you can, is it invited? I said, well, it's an invited review. I'm inviting myself to write it for you. And he kind of goes, oh yeah, well, Muscle and Nerve is really good that way. They like reviews. So we wrote an invited review. It looks like, somebody asked, oh, would you guys do this? We didn't. We volunteered. And we've done that a fair amount. And it's fun. It's a good way to learn. And again, what we did is we developed some new methods to look at the structure of collagen, mostly endomysial collagen versus paramecium collagen. And again, the take-home lesson was the endomysial collagen, the collagen right around the fibers, is organized sort of how you see here. It's kind of a mesh. The metaphor I use is, if you go to the store and buy a bag of oranges, they kind of have a mesh around them. It's just a way to keep a certain volume at a certain volume. It doesn't matter what shape it is. Same thing with the stuff around muscle cells. And it doesn't change too much with CP. But what does change is the collagen in the paramecium. We call them paramecium cables. People have identified them in the heart. I don't think I ever learned about it in school. I don't think you've probably heard about them. We think of paramecium connective tissue as kind of amorphous type 1 and type 3 collagen, but there are distinctly defined cables. You can see them here. You can look at very high magnification and see that they're regular collagen fibril cables, type 1 and type 3 collagen. We also developed a new method to to antibody label the cables and then under confocal microscopy stretch them to try to get their mechanical properties. So this is just an example, again, from Alison Gillies' work. These are cables that are running along the cell. You can see that there's a certain point on them. From left to right, these are longer and longer sarcomere lengths. And so as you stretch the cable, lots of different things happen. They rotate, they translate, et cetera. We started doing these, and again, I cut 2 years of work short. Turned out that when you do these kind of preparations, you also break a lot of the connections that these collagen cables have, and their mechanical properties are altered. So again, with Alison, I talked her into really getting into a new method that I'm using now a lot, that I love, which is called, well, it's called serial block-faced imaging. The reason it's important is all of these methods, whether it's scanning electron microscopy or confocal microscopy, require you to disrupt the tissue. This thing takes the tissue fixed in place and allows you to identify its different components. So here you see a muscle cell, muscle cell, muscle cell. This thing right here, you know, if you took a histology class or an ultrastructure class, you really probably wouldn't know that's a fibroblast. It's a little piece of an arm of a fibroblast. And then all these little dots are these paramecium cables. Here's a cable, that's kind of a nicely defined one. Capillary, 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 nucleus. And I tell you, my wife thought I was crazy when I was looking into Jan's eyes. She really thinks I'm crazy when I watch these movies of serial block face images. I just sit there and I go, oh, look at that. Oh, look, right there, right there, right there. Oh, over here. Oh, look at that. Ooh, that, see. And then, you know, I know. We end up with these 3D reconstructions. So the pink are the capillaries, the blue, look at that gigantic fibroblast. I mean, it's unbelievable. These things are huge, and they go everywhere. It's like it's like they have their feelers everywhere. It's like they're all over the matrix, keeping track of what's going on. It's like they're the dictator of the extracellular matrix. And we've been studying their biology and what they do and how they're organized. And I'll just tell you, we have also done a lot of biochemical assays in the extracellular matrix, and the only thing so far we have to tell you, and it's not all that fulfilling, but it's really good science, meaning we're doing high resolution, very high sample size stereology. You can take pictures of this, but if you publish it, I'm going to kill you. The only thing that's really different, I'll find you, the only thing that's really different is the fraction of collagen fibrils that end up in bundles changes. So the idea now to think about in terms of how collagen is organized in the matrix, there's lots of collagen, but I think there's an equilibrium between fibrillar collagen and bundled collagen. I don't know who regulates it. I know who regulates the process in terms of the enzymes, but there's got to be cellular regulation of it. But as those fibrils end up in bundles, the system gets stiff. And whereas I showed you collagen content doesn't correlate with stiffness, fraction of collagen fibrils in bundles does. So I think that's a new extracellular process that's going on that's regulated somehow that we don't understand. And if I can get funded to study it, I will. So the second problem with muscle in CP is that the collagen content is increased and the collagen bundle organization is altered. And we're in the middle of trying to understand why. And I will also tell you, there's some good lessons in here. Another lesson is when you need to change the tool that you're using to measure something new, change the tool. And if you need to invent the tool or get collaborators. But one of the things that I think is going on in our field, in CP, is we're using the same tool over and over and over and trying to make bigger and bigger conclusions, but they're just the same old tools. So if it's not the right tool for the detailed question, change the tool. Okay, and then the last topic I want to tell you about is the whole idea about all the other stuff present in the tissue. So there was evidence through sarcomere number decrease, the fact that the fibers were smaller and that the kids had trouble growing, that the stem cell population in the muscle was decreased. And Sudarshan, Dayanidhi, and I invited ourselves to write another review for Muscle and Nerve, and this is a great review. Actually, he did it, and he's a great figure guy. Isn't that a great figure? He's great at that. It's a story about how the stem cells live and develop in the muscle. And it's really interesting. And stem cells in muscle are required for growth, regeneration, etc. So it's possible to isolate the stem cells from muscle. And this is work that Lucas Smith did at the very end of his thesis. And the shocking result was that if you look at the population of different cell types, these are just examples of the kinds of cells you can isolate. The satellite cell population is dramatically decreased in kids with CP. Now, I already told you that the extracellular matrix is messed up. And so part of the reason the satellite cells could be decreased could be because they just get stuck in the matrix. But this study was pretty easy to do. Sudarshan followed it up with a very difficult labor-intensive study and basically got the same result. And so we believe that the satellite cell population in kids with CP is decreased by maybe 60 to 80%. It's really bad news, because you need stem cells to grow, you need stem cells to adapt, you certainly need stem cells to regenerate. So it has tremendous implications for all these processes and kinds of things kids have trouble with. So again, I'm not a cell biologist, but I know people who are. It's one of those things. So this is work now Margie was doing at the end of her thesis. Andy Domeneghetti, who is a true biologist, started studying what's wrong with these satellite cells. Because one of our ideas was, okay, they've lost their satellite cells. How about if we just take a little chunk of muscle, grow it up? It's very easy to do. That's actually easy to make them proliferate. And then maybe inject them back in as a cell-based therapy for kids with CP. I wrote a business plan. I had just gotten my MBA. I got investors. I was all excited. And the investors had, as the first milestone, the ability to grow and differentiate satellite cells. So I thought, well, I know I can do that. But just to humor these guys, they're the people with all the money, so I decided, okay, I'll go ahead and do it. We grow them. They proliferate. There's no problem. And, I'm getting all excited. I'm ready to go out and get the next chunk of money. And then Margie says, they are proliferating, but they don't look right. And I go, what? She goes, yeah, they're not really maturing. They're growing. They're undergoing mitosis. But, muscles go from MyoD, transcription factors, through myogenin. These guys weren't going down the developmental pathway. And in fact, just even from the picture, the top panel is CP myotubes. And we talk about how kids with CP have small muscles and they're a little bit spindly and all kind of stuff. The satellite cell myotubes are kind of spindly too. And if you measure the myotube area, it's smaller. If you measure the number of nuclei per myotube, it's smaller. So there's just a defect in their ability to fuse and grow intrinsically. And that has two important implications. The most important one is the little company that we thought we were going to form is over instantaneously. There is no company if you can't grow satellite cells. And the second thing is, the good news is development of muscle is a pretty well-defined field, and there are other factors that we could potentially use to fix the satellite cells. So Andy and I are now looking at the development of stem cells in kids with CP using biological approaches. And here the idea is, on the left-hand side, you see a typically developing couple of cells. The red is the slow myosin heavy chain. Remember, myosin is the motor. So red means these guys are developing into contractile myotubes, and they're starting to get their act together to turn into muscle. The right side, now remember, this is CP satellite cells, and they're in the exact same juice. One of the things we thought was, well, the upper motor neuron lesion is affecting the muscle's ability maybe to maintain satellite cells or grow or whatever. But if you take the cells out and put them into yummy juice, cell culture juice that's full of insulin and growth factors, certainly it's going to grow fine. It doesn't. So there's an intrinsic defect in the satellite cell's ability to mature, which is also terrible news for rehabilitation. I mean, good luck rehabbing a muscle that's got crummy satellite cells. It's just going to be a huge uphill battle. So our thinking now is, what we're doing is we're trying to develop epigenetic modifiers to adjust the way the cells develop and combine that with therapy. So Andy is now screening a variety of molecules and a variety of cell processes that are associated with development. So one of the most obvious things, if you know about cell biology, is these integrin families are families of adhesion molecules or complexes that stick cells to their substrate. And if a cell can't stick to its substrate, it won't grow. So if you look here on the left-hand side, 4 hours after growth, the typically developing kids are expressing tons of beta-1D, which is one flavor implicated in muscle, compared to CP, and that goes on even later. And they're expressing the right amount of a lot of other proteins, so it's not just that the CP kids are producing less protein. And what we've been able to do now is use various molecules. We're out there screening molecules, just like a lot of biologists are doing, screening molecules that will change the way these cells behave. And the hope is that we will be able to use already approved, FDA-approved drugs that are epigenetic modifiers to work in this field. The epigenetic drug field is huge. It's $1 trillion industry, and it is huge in cancer. So our field is not a $1 trillion industry, and we're always mad at that, but we can use the stuff that they're doing because it's still the same biology. And we think that there are some drugs that have failed cancer trials that will pass our trials because they've already proven to be safe in people that we will be able to use. So we'll see how that goes. So the third thing that's wrong with muscle in CP is that the satellite cell number is dramatically decreased and it's epigenetically modified. That is also a problem. Do I have?
Yes.
Okay, I got one last thing to tell you. This is work that Sudarshan is doing, and I think it's really important because I told you that the contracture and the satellite cell changes and the sarcomere length changes were uniquely human. And that's one of the reasons a lot of the mouse, rat, cat, dog data haven't helped us much. So this is the way we're thinking. I already showed you that the satellite cell number is decreased. Sudarshan showed it, Lucas showed it, and I believe it. And I also showed you in a bunch of different studies that sarcomere number is decreased. And just like the collagen stress data, the question is, are they causally related? Are they just two things that are happening, or do they go together? Well, it's pretty hard to study. But we collaborated with a group in Kentucky who made a mouse model that they were using for other purposes, but we stole it from them. That's called collaboration. We collaborated with them and got their mouse, and it's a really special mouse. It's a mouse that's walking around happy-go-lucky, living a normal mouse life, you know, eating cheese. And then all of a sudden you feed it tamoxifen, and the tamoxifen does a chemical reaction that ends up killing its satellite cells. So you can make a mouse that doesn't have very many satellite cells, even once it's an adult, and do a bunch of experiments, and I'll show you one of them. This is also from Sudarshan's work. Basically, the idea is shown here. You immobilize mice in a plantar flexion position. When they are immobilized in plantar flexion, they lose sarcomeres. You treat them with tamoxifen, and then you let them walk around. And the really encouraging finding is, when you let them walk around, they end up, unlike normal mice will, you know, the contracture will work its way out. The mice who have lost their satellite cells end up with plantar flexion contractures. So, I mean, if I'm a therapist, I'm all over this result, right? Because now I can be a mouse therapist, and I can ask the questions. You know, all the questions we ask about intensity and duration and frequency and all these kind of things, you can ask them on some of these animal models, and we're very hopeful that this will allow us to do some direct testing. So, I think I will summarize. Whoops, sorry. Not going? There we go. Muscle contracture represents a complex, I hope I've convinced you about that, and I think it's uniquely human muscle adaptation. The fact that passive muscle mechanical properties are altered to a greater extent than active mechanical properties is bad news, because we know a whole lot more about active muscle. That's what all the genius muscle physiologists have been doing for the last 50 years, is active muscle contraction. The passive stuff is not very well understood. CP muscles have trouble growing, either in length, sarcomere number, or in width, fiber size. And curative therapies are hampered currently by lack of knowledge in several specific areas, like the structure and function of the extracellular matrix, how it's regulated. Remember I told you who's in charge of making collagen fibrils, and then who's in charge of assembling them into cables. And of course, satellite cell development and function is a gigantic area in the muscle disease world, and that's an area that we should be exploiting. So thanks very much for your time, and I'd like to acknowledge funding sources and would be happy to take comments or have questions. Thank you.
Okay, thank you very much for your very interesting lecture. As always ends research with the remark, further research is needed. But we're coming closer to the problem, that's for sure. Are there questions from the audience? In the beginning, no questions. Well, I have some questions. It's very interesting. There's one thing about the satellite cell problem, that they are different. Well, when a baby is born which will develop CP, the muscles are not stiff. They are even hypertonic. So it's a feature which develops in time. And I'm wondering, is it an option that, well, the basic problem was a period of hypoxic period which damaged the brain, but which also is present in the whole body. We look mainly to the brain, but actually it occurs in the whole body. Is it an idea that the hypoxia also damaged the satellite cells?
It could easily be. So right now we have data spanning maybe a 10 or 15-year age range, and there's no correlation on our data, which are very noisy. There's no correlation between age and satellite cell number, or satellite cell density. So I don't think, I mean, at least the way I'm thinking of it in my brain, I don't think the kids have sort of their normal complement of satellite cells, and they're slowly going away over the multi-year period. I think either during development, the cells never stayed behind, because that's what happens during development. Myoblasts fuse, but if you were left behind, either they didn't leave them behind because of a hypoxic environment, or there's something that the central nervous system is actually telling the muscle during development about stem cells. Like, honestly, that topic, how the brain and central nervous system could affect muscle development, is totally untapped. So it could be any of those things. Either the muscle didn't develop appropriately, or the signals from the central nervous system are allowing the satellite cells to senesce pretty quickly. It's a really interesting topic.
Okay. I see some fingers.
I can't see, I'm sorry.
Yes?
Oh.
On the left, and then the lady on the right. Yeah, on the left side.
Sol, I'm from Oslo, Norway. I had a question about the satellite cells as well. You had trouble growing the satellite cells. Could it be an idea to, with a unilateral CP, to grow it from the good side and use it on the bad side?
So far we haven't been able to get biopsies from the good side. It's probably worth talking about. It's a great idea. So far we haven't been able to do that. We either have typically developing muscle or CP muscle from the contracture side.
Okay, and there was.
Hello, thank you for a very interesting talk. We were thinking about what does it mean for the restitution that the satellite cells are so low, the amount of satellite cells.
What does it mean for in terms of therapy and this kind of thing?
Yeah, and the restitution for after training.
Sorry, the restitution of what?
After training.
Oh, so the satellite cell literature is pretty clear that satellite cells are absolutely required for muscles to regenerate. And there are, in terms of like exercise-induced injury, there is injury to muscle cells that upregulates satellite cells that rebuilds muscle fibers. So the implication would be after like intense training, that the intense training may not produce strengthening because that's part of this sort of regenerative strengthening response. I don't think anyone's ever shown the training piece even in mouse models. Do you know, Sudarshan? Do you know if they've shown it in exercise?
I mean, the only thing which is shown is that you can increase satellite cell numbers, but with training, if you do endurance exercise. If you do endurance exercise, you can increase the numbers, but not when it's that low. Typically,
So as long as there's no direct experiment, what I would say is that the implications are that the direct implication, without really understanding this phenomenon that very well, is if a muscle is lost, let's say 70% of its satellite cells, it'll be very hard to strengthen it by exercise. It'll be very hard to stretch it and have sarcomere number grow by, casting and this kind of stuff. And it will end up producing roughly lower forces because the fibers will be smaller. That's why I said when we found out that the satellite cells themselves were lower and not normal, it was really bad. It was really bad news, because for a while there in the CP world, people realized that they just weren't driving the muscles hard enough. So there was a big push on intensity and all that kind of stuff. And then people did good experiments with EMG and force plates and all that stuff and figured out, we're exercising these kids very intensely and they're still not growing. And now it sort of makes sense to me. It's sort of like they can't grow. So it's almost like we have to unlock the potential of the stem cell niche within the muscle and then do all this therapy.
Okay. Is a last question?
I have a couple of questions. Is this mic on?
I think you only get one.
You get a time for one.
The second one is $20.
Oh, boy. So it really has to do with, were these results across the board with all different types of CP? And to follow up with that question then is that the thought that the preemies are having problems with it's a stem cell issue rather than a strictly hypoxic issue with struggles with their oligodendrocyte precursors. And is there a role there along with these satellite cells?
I have no data on the preemie question. Almost all these kids were spastic diplegic kids. So.
Most spastic dying are preemies, right?
Yeah, but I'm saying I don't have enough preemie versus non-preemie to differentiate between them or among them.
Thank you.
Cool.
Okay. Well, thank you very much for your very interesting talk, and to be continued. Okay. And now it's... And it's time for the tea break.